Okay, let's get started. All right, we're going to begin this lecture with a clicker question. So this is from last time, and the question is, why is the membrane potential so close to the equilibrium potential for potassium? And the possible answers are A, potassium has a much larger concentration gradient across that membrane than sodium. B, the cell is much more permeable to potassium than to sodium. C, potassium and sodium play only a small role in the membrane potential. D, potassium ions have greater charge than sodium ions. And E is all of the above. Let me start the software. Okay, so take a minute to think about that. Okay, give me your final answer to this question. Because I will turn it off right about now. Give me your answer. Okay, so most of you correctly remembered from last time that that's not the correct answer. I don't know why it highlights like that. Okay, so we cross that off. And most of you remember the correct answer is B. Okay. So about 80% of you got the right answer. All right, so we were talking about permeability. This is in the context of basic principles that apply to excitable tissues. And excitable tissues include neurons and muscles. Okay, and what we learned was, first of all, we have these electrochemical gradients. They create situations, especially when facilitated by the action of sodium-potassium ATPase, where we create an imbalance of the concentration of sodium and potassium across membranes. We create voltages. And those voltages can be modeled if we know the concentrations of the key ions across the membrane. And they're also subject to change. And this is where we left off. So how do they change? They change because membranes, the cellular membranes of these excitable tissues have the capacity to change permeability. And we can model exactly how permeability relates to those voltage changes using the Goldman equation, which we see here. We have this example of changing permeability, seeing that reflected in the membrane potential. How does the permeability change? Well, we talked about how there are these transmembrane proteins called channel proteins, and they're essentially these liquid-filled pores that are specific to a particular ion. And one class of them is they're just open all the time, like the potassium leak channels. But then there's this other class of gated channels. And they can be gated in these three different ways. Either they bind to a chemical called a ligand. Those are the ligand-gated channels. They're pulled on and they open up and increase permeability as a function of tension in a cellular membrane. Those are mechanically gated channels. And then there are voltage-gated channels where the membrane the voltage of the membrane potential, influences whether or not that channel is open or shut. So in response to this question that we posed last time, how does the potential change? That's facilitated by changes in the permeability to an ion, and this is facilitated by gated channels. So just to step back to our broader context for part one of this course, where we're covering epithelia, muscles, and nerves in detail, we're now on these excitable tissues. And now that we've sort of set up with those basics of changing permeability and influencing the membrane potential, let's start talking more explicitly first about neurons, and then we'll get into muscles. All right, so here's my cartoon neuron. That gray or beige dot is the nucleus of the cell. And there are two basic components to a neuron. There are the dendrites. The dendrites' main function is to receive signals. We'll talk about signals being received from sensory organs or from other neurons, but those are the collecting parts of the cell that branch out and receive information. And then a decision is made about whether or not that collection of information is sufficient for a signal to be passed on down the other portion of the cell called an axon. And then the axon terminates. You might have something like a muscle cell or maybe another neuron there. But there quite often, in the context of what we'll talk about, there will be a chemical synapse and a release of neurotransmitter at the end of the axon. Okay, so we'll talk about those actions at the end. All right, so let's start this presentation by focusing on the axon. And then once we sort of lay out the basics of how the axon works, then we'll go back and consider the dendrites. Okay, so the first question is, what is so special about the axon? And in this part one of the course, a lot of our answers to these questions are going to be in the form of, well, the membrane has these sort of wonderful properties that endow this portion of the cell or this type of cell with these capacities. Okay, so the axon transmits information really well. What is it about the membrane that endows that part of the cell with this capacity to do that? So we're going to very often consider sort of thought experiments. We're going to step through experiments in a very schematized way, but they're similar to what you would do if you were an experimental physiologist, in this case a neurophysiologist. And if you were to perform electrophysiology on this neuron to interrogate, as was originally done by Hodgkin and Huxley, to interrogate the properties of the membrane in this part of the cell, then we would do that with these microelectrodes, which I've described. And so here we have a voltmeter with a microelectrode that has pierced the cellular membrane of the axon, and then we have an extracellular microelectrode that's outside of it. So this is our reference electrode, this is our recording electrode, and the recording electrode tells us the voltage at that point relative to outside the cell. And you would perform this experiment with the cell bathed in salt water, something close to physiological saline. These experiments were originally done on squid axons. Why squid axons? Well, it turns out that squids have exquisitely large axons. And so when you're a physiologist, you always look for animals that have a big piece of the thing that you're interested in, because it just makes it much easier to work with, especially when you're developing novel techniques for these kinds of recordings. So that's going to be our measurement. And so we're going to be monitoring the membrane potential. That's what we see down below, where we have the membrane potential as a function of time. And I've already put the resting membrane potential, which is about minus 70 millivolts right there. Now, you don't generally have to memorize much in this class, but it's good to be familiar with some numbers, okay? So it's good to remember that minus 70 millivolts is about the resting membrane potential of our sort of generic neuron that we'll discuss. It's just kind of a useful reference for changes in the membrane. And as we've already established, we can change that membrane potential if we can change the permeability of the cellular membrane to particular ions. And so up top, we're going to have permeability. And we're going to track the permeability of the membrane to sodium. And so we see up above, we've got a flat line of sodium. And down below, we're just stuck at the resting membrane potential. But if we can monitor both of these properties, then what we would find is that if we have an instance where the membrane potential starts to rise, we would see that reflected in the permeability for sodium, something like that. And if this were to continue on so that the membrane potential exceeded a value of minus 55 millivolts, or thereabouts, then what you would find is that this rise, and this is, by the way, a spontaneous measurement, okay? We're not experimentally manipulating anything. The giant axon of the squid will just do this on its own, okay? So this is just an observational data set. And if that neuron gets to that value of minus 55 millivolts, then we see that there's an inflection in the slope of the change of that membrane potential. Okay, it's not drawn very well here by me, but if you focus in, right here, you can see that the slope is gradual, and then it becomes steep. Okay, right around negative 55 millivolts. And then you also see the same kind of change in the curve if you were to track the sodium permeability. Okay, so they're correlated. Another thing we could do in this situation is monitor the concentration difference of sodium on the inside of the cell and on the outside of the cell. We could use an equation to calculate the equilibrium potential for sodium. That equation would be the Nernst equation. The Nernst equation will tell us, of course, what the membrane potential will try to approach if we make the membrane really permeable to that ion. So that's a useful reference to add to this graph, okay? That's where the equilibrium potential for sodium is. It's less important to remember what that particular number is. It's just important that it's a really positive number, okay? Plus 60 millivolts. So this is what we would expect, given what I told you about the Goldman equation, all the stuff in the last lecture, that if you make the membrane permeable to sodium, like if you make it really permeable to sodium, then the cell membrane is going to move towards that equilibrium potential for sodium. So given that, this is not just a correlation, it suggests that there's causation here, that this permeability change is driving this, what we call depolarization, a really rapid rise in the membrane potential. And then if we continue on through time, we see that this drops, and so does the permeability change for sodium. All right, so these spontaneous, rapid changes in membrane potential up and down are what we call action potentials. And action potentials you only find in the axon. You don't find them in the dendrites. And they have some really fantastic properties if the focus is on information transmission. One is that they're all or none, okay? So you've got essentially a binary signal, okay? Either you're generating an action potential or you're not generating an action potential. Now, we're going to talk a lot about action potentials. We have them not just in axons, but also in muscle fibers. And so there's a whole terminology which we cannot dispense with. As much as I like to get rid of jargon, these are some terms that we actually need. And so the term that I just mentioned was depolarization. That is, if you have a rapid, or if you have any rise, it could be rapid or slow. But if you have a rise in the membrane, then you say it's depolarizing. Okay, it's going up. Question?
Yeah. So would polarization be a fall?
Say again?
Would polarization be a fall?
Be a fault?
A fall, like a fall in membrane potential.
So if you have a drop below the resting membrane potential, then we call that hyperpolarizing the membrane right here. Now, if you're already at an elevated state and you're returning back to the membrane potential, that gray line, we call that repolarizing. So you've got depolarizing, hyperpolarizing, repolarizing. Sure. I mean, a way of thinking about the terminology is that at rest, it's polarized. You know, you're not at a zero membrane potential. And then you're sort of undoing that polarization, so you're depolarizing it. And then we're going back to that polarized state, so we're repolarizing. And you can have it even more polarized than rest, and that's hyperpolarizing. Okay, so that brings us to another clicker question, which is, what causes this repolarization? Now, you might at first think that it's going to be driven by the drop in sodium permeability, but notice that is not one of the options. Okay, there is something else that is driving the repolarization. So is it A, a reduced permeability to chloride, B, an increased permeability to potassium, or C, the action of sodium-potassium ATPase? Okay, so what causes repolarization? Okay, give me your final answer. All right, about 3/4 of you got the right answer. So it's not A. We haven't really talked about chloride yet. It turns out that chloride, well, we'll talk about chloride later. So right now, chloride's not really in the mix. And so that leaves both potassium and sodium-potassium ATPase. Now, both eventually have the potential to do the same thing. If you make the membrane permeable to potassium, it's going to go down because we already established before that the equilibrium potential for potassium is a really negative number. It's negative 90 millivolts or thereabouts. And in fact, we have it there on the graph. So that's the equilibrium potential for potassium. So if you make the membrane permeable to potassium, then the membrane is going to tend to go towards that. Sodium-potassium ATPase also acts to establish the resting membrane potential, but it does it not on these time scales. It does it relatively slowly. So if the goal is to create rapid change in the membrane potential, then you want to do that by changing the permeability that is manipulating the Goldman equation in order to rapidly achieve that change. So it turns out that B is the correct answer. And so if you track the permeability changes of potassium, and that's where we'll go back to that graph up top, that you see that it looks something like that. That is, it begins to rise at the same time as we have the rise in sodium, which is shown in purple. So the green curve, I don't know where my K went, but the green curve is for potassium, and the blue curve, purple curve is for sodium. So they both rise at the same time, but the rate of the rise is slower for potassium. So as a consequence, there's a delay. Sodium permeability has the rapid rise that drives the membrane towards that equilibrium potential. And then very shortly thereafter, you have the rise in potassium permeability, which then drives the repolarization of the membrane. So this decline in the slope. Does that make sense? I mean, very quickly in these explanations, we're going from here's a simple membrane, here's Nernst equation, here's Goldman equation. Next thing you know, we've got gated channels and everything. It gets complicated rather quickly. Yeah?
I'm trying to comprehend how the increased permeability of potassium itself, like the general thing, what it does.
What does, okay, so if, so just sort of when in doubt, go back to the, let's see here. Go back to the Goldman equation. And well, so again, this is why I sort of emphasized last time how, you know, having an intuition for what the Nernst equation tells you and what the Goldman equation tells you is key. So the Nernst equation tells you what the equilibrium potential for an ion is. All right, so that gives you the plus 60 for sodium, the minus 90 for potassium. And then if you change permeability for a particular ion, you're going to drive the membrane towards that equilibrium potential. Now, the rate at which that happens depends on the magnitude of the permeability change. And here we don't talk about that in specifics. We just say, okay, we're going to make the membrane permeable to a particular ion. So you make it permeable to sodium, it's going to go towards plus 60. If you make it permeable to potassium, it's going to go towards minus 90. Okay? So in an action potential, it goes up because initially you have this big spike in sodium permeability. Then it drops because very quickly soon thereafter, you have the rise in potassium permeability. So that brings it back down towards, it approaches the minus 90 millivolts because that's the equilibrium potential for that potassium. Does that make sense? Okay. Yeah.
Is the permeability of potassium increased by just like modifying one membrane protein, or is it just like a...
Okay, so yeah, you're very good at sort of setting me up for the next topic. And so that is... Okay, so we're going to talk about what drives the permeability changes that we just documented in the graph. Okay, what are the molecular mechanisms? And so this was only established many decades after the initial electrophysiology and the models that were established by Hodgkin and Huxley using the squid giant axon. And so this is all what we know now about how this is facilitated. So first we'll talk about sodium and then we'll talk about potassium. So initially, we have these gated channels for sodium. And these turn out to be voltage-gated channels, not ligand-gated, they're not mechanically gated. These voltage-gated sodium channels at rest are shut. Now, I have to warn you here. In my little cartoons, I'm going to focus on one particular channel. You just always have to bear in mind that there's a large population of these channels, okay? So there are thousands upon thousands of these things. And at any particular moment, some might look like this, some might look a little bit differently, okay? This is important when we talk about sort of the time scale of things because we'll see these relatively gradual inclines in the membrane potential, permeability, things like this. And that's because you have progressively more and more channels opening up and closing, okay? It creates some damping to the signal. It's not a sort of an immediate increase like you would expect if there was only one channel that mattered. This is sort of like when I was explaining diffusion. We talked about a particular particle and how it behaves, and then we talked about the population. Okay, so the individual looks something like this. This is sort of your average channel in that population. At rest, it's going to be shut. And that means it's going to keep the sodium from traversing the membrane and keeping the permeability relatively low. Okay, that makes sense. This one property of these voltage-gated channels is that they have a threshold voltage where if the membrane potential exceeds that voltage, they change their state. They open up and they elevate the permeability to sodium. Okay, so at threshold, we see that the channel, this average channel, opens up, and that allows the sodium across the membrane. And these channels only allow sodium across the membrane. Okay, so above threshold, we're in this situation where a depolarization, that is a rise in the membrane potential, has driven an opening of channels. Some portion of this population of channels have been triggered. And what do? They let sodium across the membrane, thereby driving the membrane potential towards the equilibrium potential for sodium, which is a really positive number. In other words, they trigger other voltage-gated channels to also open up. And so what you have is a situation where there is a positive feedback loop. Okay? You have channels opening up, which further open up more channels, which in turn tend to open up more channels. And there aren't many physiological situations where we have positive feedback loops. I'll cite like 3 of them in the whole course. But one characteristic of positive feedback is that they drive a rapid change in state. And that's indeed what you see with an action potential. It's a very, we'll talk about the timescales, but the initial portion of an action potential takes 2 milliseconds. Okay, it's really fast. Okay, so another characteristic of positive feedback loops, and the reason why we don't see too many in physiology, is that they tend to be uncontrolled, right? They have this very rapid change, and if they're never corrected or shut off, then they can never kind of reset and get back to normal. And in general, homeostasis is about being normal, having a very stable environment. So the feature of this, this voltage-gated channel that allows for a shutting off is a portion, this is a domain of the protein itself that shuts it down. And that domain is called the inactivation gate. And the inactivation gate is engaged when the membrane potential gets up to a certain very positive value. So it's triggered to close by the membrane potential itself. So we have sort of 2 ways that this voltage-gated channel is triggered by voltage. One is to open up what's called the activation gate, that's the initial gate, and then we have a closure of the system by the inactivation gate. Okay, so once that is closed, then the sodium can no longer traverse the membrane. Okay, so the voltage-gated channel for sodium is more complicated than the one for potassium. So potassium, once you absorb that, the potassium channel is easier to understand because it only has an activation gate. So again, sodium's higher in concentration on the inside of the cell, and so it wants to move down its concentration gradient to leave the cell, and that's prohibited by the activation gate when the cell is at rest. But once the threshold is reached, there's the same threshold as the voltage-gated sodium channels, then we have an opening, and then it closes also according to the membrane potential. So we have a little cartoon for that. It opens up, potassium rushes out. That's going to be driving the repolarization. And then once the membrane is back to its approximately its resting membrane potential, then it's going to shut down, and we see that there's going to be a decline in permeability right here. Okay, so note that the membrane actually will do this undershoot where it hyperpolarizes just momentarily, and then eventually it goes back to rest. Okay, that hyperpolarization is because the membrane is still being driven towards the equilibrium potential shown as the dotted line there for potassium before we have the shutting down of the membrane to that ion. Okay, so we have these changes in permeability. Initially, for potassium, we have a closure of the activation gates. At this point in time, the activation gates are also closed for most of the sodium channels. Then, this is just to summarize, then we have the depolarization which activates both channels. the activation gates open essentially more slowly for the potassium. Okay, that's why we have a relatively slow rise compared to sodium. All right, so sodium activation gates open quickly. The potassium activation gates are relatively slow. It's still really fast, but slower than sodium. And so if we move on to the repolarization, at this point in time, the sodium channels have had their inactivation gates closing, the permeability for that ion, and the potassium voltage-gated channels are open and keeping a relatively high permeability for potassium, shown as that green line there. And then when the membrane is hyperpolarized, then we have a return back to rest. And at this point in time, both the sodium channels and the potassium channels in general are closed. Okay, does that make sense? Okay, so those are all of the details of how two voltage-gated channels drive the permeability changes that facilitate an action potential. Question?
Is there going to be a slide where all that's summarized, when at the different states, when one gate is open, one gate is closed?
I just gave that slide. So this does illustrate how my slides work better as video than statically. But fortunately, I did press record, so it's all captured. Okay, so this is a lot of what makes axons really adept at transmitting signals, because action potentials not only are rapid when you're looking at one little spot along the axon, but that signal also propagates down the length of the axon quite rapidly. Okay, so what is so special about an axon? Well, it transmits these signals, these action potentials. The other thing that makes them special is that they do it only in one direction, at least in our bodies. Some animals have bidirectional neurons. You see that like in jellyfish and other animals that have nerve nets. But in our bodies and in vertebrates in general, axons transmit information just one way. And they do it quickly. Greater than 300 miles per hour, if you want it in those units. And they also have this capacity of transmitting information to neighboring cells. Okay, so we talked about how action potentials are generated, but we haven't touched on how any of these other properties are possible. What is it about the membrane that gives an axon unidirectional transmission? How is it achieved quickly? And how does it transmit that information to neighboring cells? All right, so what we're going to do is another physiological experiment, or thought experiment, where we've got our recording electrode, but now we're going to manipulate the system. We're going to add a stimulating electrode, and this was, again, classically done by Hodgkin and Huxley. And so we pop it right there. Stimulating electrode is closer to the cell body. And so upstream of what you would expect for information transmission, because when I say that the signals are transmitted in the cell in one direction, that direction is from the cell body up here down towards the axon terminal down here. And the manipulation that we're going to perform is we're going to simulate what happens when those voltage-gated channels are activated. Okay, so we're going to drive a depolarization. And what you can do is if you have in your stimulating electrode a bunch of sodium ions, then you can essentially manipulate the axon as though it's creating its own depolarization. So there's our microelectrode zoomed in across the cell membrane. We're going to inject sodium ions, and we're going to experimentally depolarize the membrane right at that one spot. Okay, and we refer to that as injecting current to change the membrane potential. Okay, so let's consider what happens when we do that. No cell phones, please. Okay, so here we have a stimulus voltage that we're going to impose. We're going to simulate a situation that is similar to an action potential, right there. And let's monitor what happens for the membrane potential downstream of that stimulating electrode. Okay, so initially at time 0, we see we haven't yet injected it, so that we see nothing. But then we're going to drive a depolarization downstream. And we can see that it's slightly delayed in time. And if we do it strongly enough, if we have a high enough concentration of sodium, we put enough sodium into the system, then we're going to drive the membrane right here to threshold. And it will undergo its own action potential right there. Now, let's start to add time. Okay, down below you can see we've got time in milliseconds. And when the whole action potential, the whole process is done, we've gone 2 milliseconds in time. Okay, pretty quick. All right, let's do a slightly different experiment. Let's, instead of giving it just one excitation, we're going to give it 2 spikes like that. And let's just see what happens. Okay, so in response on our recording electrode, we see exactly the same thing. Okay, the action potential looks just like it did a moment ago. There is no effect of having that second pulse, that second depolarization. The axon is in fact insensitive to a second depolarization. And you could repeat that experiment for any moment in that gray period of time, and you get exactly the same result. You would see exactly the same action potential downstream where at the site of our recording electrode. Okay, what if we move our stimulus outside of that 2 milliseconds? So here we've got small depolarizations. They're just enough to drive an action potential at the site of our recording electrode. And we see same thing. Okay, again, with a small stimulus outside of that 2 milliseconds. So we're talking about between 2 and 4 milliseconds. In this period of time, if we have a small stimulus, then we're going to see exactly the same result, same action potential. But if instead we give it a stronger stimulus and we're outside of that 2 millisecond window, then we start to see something different. Okay, finally we're getting a different result. And notice that the magnitude of that action potential is just as much in the second spike as in the first spike, right? Because action potentials are all or none. Either you've got an action potential and it's strong, or there's nothing. So in this period of time, which we call the relative refractory period, a strong depolarization is required in order to overcome a higher threshold. Okay, now let's go back and think about this in terms, well, there's 2 reasons for this. First of all, early on anyway, right around the 2 milliseconds, we can see that the membrane is still hyperpolarized, meaning it's at a more negative value than the resting membrane potential. So in order to create another action potential, we're starting from a more negative point, so we have further to travel. So that's one reason why the axon is less sensitive in the relative refractory period than it is at rest. The other problem has to do with the fact that we're dealing with a population of voltage-gated channels. Now, I mentioned that we have the inactivation gates shutting down your average voltage-gated channel. The voltage-gated channels that were excited early on in this whole process, that is when the first action potential began, those are going to start resetting and opening up their inactivation gates and being ready to accept another depolarization early, right? The early ones are going to be reset early, and then the later ones are going to be reset later. So there's a whole population, and right at 2 milliseconds, there are very, very few voltage-gated channels to be activated, sodium voltage-gated channels. As you move through time and you get closer and closer to 4 milliseconds, you have more and more of those voltage-gated channels to recruit to create another action potential. So actually so what's being drawn here in this dotted line here is the threshold during the relative refractory period. So as a consequence of having fewer channels to excite, then effectively the membrane potential has to be higher than it would have been at rest. Okay, at rest it's minus 55 millivolts, and in the relative refractory period, it's higher. So actually, this is drawn a little bit incorrectly because this threshold actually goes down over time. So here it's drawn as a flat line, but in fact, by the time you're at 4 milliseconds, it will have returned back to minus 55 millivolts, the sort of standard threshold value. Does that make sense? That's a little confusing. Okay, so first of all, there's the observation. If you try to activate a neuron within 2 milliseconds of when an action potential has begun, you get nothing. Okay, it is impossible to activate the axon in those 2 milliseconds. When you go from 2 to 4 milliseconds, which is what we have in gray here, then you can do it. But you have to give it a sort of more strong depolarization in order to create a second action potential. Now, the explanation for why that's true is we're starting from a more negative number, and then the other thing is that you've got fewer voltage-gated sodium channels to create that second depolarization. Okay, so it's less sensitive. That's why you need to give it a higher depolarization. Any questions about that? Yeah.
How, like, in terms, like, can we quantify how strong the stimulus needs to be?
How strong does the stimulus need to be? It's very well characterized and quantified, but we're not going to get into it. Yeah. Mm-hmm.
Just to clarify, in the chart that you choose the memory potentials, are those spikes actual potentials or
the spikes down below, the membrane potential recordings, those are action potentials. Up above, there are stimulus voltages. They're sort of like action potentials, but we're imposing those. And so they're experimental potentials, but they don't have the shape of action potentials. They're not all or none. We can just sort of dial in whatever we like there.
One more question.
Mm-hmm.
For the dotted line for the second action potential, that's when it goes back, like at the end of, like, say, hyperpolarization, it goes back to like negative 55, right? Like it goes back to negative 55 to the threshold level, right?
The threshold goes back to negative 55. So at rest, our threshold is going to be negative 55 millivolts. Yeah. Mm-hmm.
Just to clarify, why is there fewer voltage-gated sodium channels? Or what if you could have caused it? I just really don't get. I mean, I guess.
Let's talk at the end about that. Okay. All right. Cool. Okay. All right. So the period of time from 0 to 2 milliseconds, when you can't create a second action potential, that's known as the absolute refractory period. Okay. So 2 to 4 is relative refractory period. 0 to 2 is absolute refractory period. And so our explanation is that almost all of the voltage-gated sodium channels are in this state. That is, the inactivation gates are closed. They're sort of out of commission, not capable of creating a depolarization. Whereas when we look at the relative refractory period, we at least have some voltage-gated channels that are reset and ready to go again. So just to summarize in the refractory periods, we have the absolute refractory period. It's in the first 2 milliseconds of an action potential. We cannot get a second action potential when that occurs. And it's caused by the inactivation gates of the voltage-gated sodium channels. The relative refractory period is from 2 to 4 milliseconds of the initial action potential. And we can see that an action potential is possible with a strong stimulus, and the reason why you need that strong stimulus is because you're starting from a more hyperpolarized state, and also you have some of the voltage-gated channels still in the process of resetting. Okay, so that's kind of a curious property. So it's critical if you're going to transmit information that is encoded by action potentials that the cell be able to reset in order to receive another action potential. That makes sense, right? Because essentially your throughput for the system is going to depend on how quickly the cell can go back to rest. And these refractory periods are sort of almost like a byproduct of that phenomenon, that sort of functional need that we have to get the cell back to rest so that we can receive another signal. but it turns out that they also endowed the cell with one of its other sort of magical properties. So let's consider the transmission and the direction of transmission given what I just told you about refractory periods. All right, so we've got our schematic axon. We're focused on it. Up top is where we're close to the cell body. That's where a signal is going to be originating from. At this particular moment in time, we have a region of the axon that is depolarized. Okay, that's where the action potential is happening. And as a consequence, sodium is rushing in. It's depolarizing the cell in all adjacent areas, right? This is chemical diffusion. It's not directional. Once it's inside of the cell, it can diffuse in any which way. The membrane that is next closest, that is further along the axon, is going to be deactivated by that influx of sodium, right? So what we can see is that now we have a new region, and the leading edge of our action potential has moved down towards the axon terminal. In the wake of that depolarization, we have a region of the membrane that just went through its action potential, and since it's a moment later in time, is now going to transition into the absolute refractory period. Now what that means is that as sodium rushes into the cell in this new region that's being activated, we have the green region that is now going through the absolute refractory period being insensitive to this influx of sodium. That means it's not going to be activated again by the sodium that's entering into the cell. Instead, on the other end, as sodium moves into the membrane, this membrane is at rest and it can be activated. So we have the action potential move along. And again, the region that was just activated transitions into the absolute refractory period, is insensitive to any more sodium, and can't be reactivated. So as a consequence, the refractory period prevents an action potential from reversing direction. Okay, so it's not just sort of a timing thing and an encoding property, but it also endows the membrane with the capacity to send signals just one way. So let's perform another experiment where we put a stimulating electrode right here and we inject some current to depolarize the membrane. Here's a clicker question. What would happen if a stimulating electrode created a depolarization in the middle of an axon? Okay, this is unlike what I just mentioned where we had something start towards the cell body. In this case, we're going to stick our electrode right in the middle. What do you predict would happen if you did that? So A is an action potential would travel both towards the axon soma, that is up on the slide, and the terminal, which is down. B, an action potential would travel towards the soma, that is just up. C, an action potential will travel towards the terminal, just down. Or D is an action potential would not travel if stimulated at the axon. This is a vertebrate, yeah. Okay, so you're going to have to give me your final answers on this one. And this is a hard question, but we're just about done. Okay, final answers. Okay. All right, so about half of you got the right answer. So let's consider what would happen. So we inject the sodium. It depolarizes the membrane right at that spot. Okay, but it's also going to diffuse in both directions. Now, at rest, both adjacent regions can be activated. So the sodium can activate both parts of the membrane. So it is true that when you observe the action potentials, they always start at the soma, but it's not because there's any polarity to the membrane necessarily. It's just that they always start at the soma and then they travel the other way. So the correct answer is A. So I'll see you on Wednesday.